What if a virus could cause severe hemorrhagic fever, multi-organ failure, and mortality rates approaching 90%? Welcome to the world of the Filoviridae family. The family Filoviridae contains some of the most feared viruses in modern medicine. Although outbreaks are relatively rare compared with influenza or coronavirus infections, members of this family can cause severe viral hemorrhagic fever with high mortality rates. The two most important human pathogens within this family are Ebola virus and Marburg virus. Filoviruses are enveloped viruses with a negative sense single-stranded RNA genome. Their name comes from the Latin word filum, meaning thread, because of their distinctive filamentous appearance. Unlike many viruses that appear spherical under the microscope, filoviruses often resemble long, flexible threads, loops, or curved filaments. This unusual structure makes them among the most visually recognizable viruses in the world. Filoviruses are zoonotic pathogens, meaning they originate in animals before infecting humans. Current evidence indicates that fruit bats serve as the primary natural reservoir. These bats can harbor the viruses without developing severe disease, allowing viral circulation in nature. Human infections are believed to begin after contact with infected wildlife, contaminated animal tissues, or environments where infected bats are present. Once human infection occurs, transmission can continue through direct contact with blood, saliva, vomit, urine, feces, semen, or other bodily fluids from infected individuals. Unlike airborne viruses such as measles, filoviruses generally require close physical contact for transmission. This explains why outbreaks often occur among family members, caregivers, and healthcare workers when proper infection control measures are not implemented. The most famous member of the family is Ebola virus. Several species of Ebola virus exist, including Zaire Ebola virus, the species responsible for the largest recorded Ebola epidemic. That outbreak infected tens of thousands of people and highlighted the devastating impact of emerging infectious diseases. Marburg virus is closely related to Ebola virus and causes a clinically similar disease. It was first identified in 1967 during outbreaks among laboratory workers in Germany and Serbia who had been exposed to infected primates imported from Africa. After entering the human body, filoviruses initially infect immune cells such as macrophages and dendritic cells. The viruses then spread through the bloodstream to multiple organs, including the liver, spleen, lymph nodes, and vascular system. One of the most dangerous aspects of filovirus infection is the dysregulation of the immune response. The viruses interfere with normal antiviral defenses while simultaneously triggering excessive inflammation and vascular damage. Symptoms usually begin with fever, headache, fatigue, muscle pain, and weakness. As the disease progresses, patients may develop vomiting, diarrhea, abdominal pain, and severe dehydration. In advanced stages, internal and external bleeding may occur, potentially leading to shock and multi-organ failure. Filovirus infections are acute diseases and do not establish chronic infections. However, survivors may experience prolonged complications, including joint pain, eye problems, and persistent fatigue. Historically, treatment consisted mainly of supportive medical care. However, major scientific advances have occurred in recent years. Several monoclonal antibody therapies have demonstrated effectiveness against Ebola virus disease. In addition, licensed vaccines are now available for protection against Zaire Ebola virus. No widely licensed vaccine currently exists for Marburg virus, although multiple candidates are under development. Today, the Filoviridae family remains a high priority for global health agencies because of its epidemic potential. Understanding these viruses helps strengthen outbreak preparedness and improve future responses. The Filoviridae family illustrates how viruses emerging from wildlife reservoirs can cause devastating human outbreaks. Continued surveillance, vaccine development, and strong public health responses remain essential for limiting the impact of these extraordinary pathogens.